Welcome back. I am Aditya Jyoti Boshak and today I am going to begin cloning experiment. So, I have already isolated cDNA from the spleen of mouse, and today's experiment, my objective is to amplify a particular gene of interest. So, as you can understand, the spleen of mouse or any organism contains hundreds of different genes. My research work dictates the requirement of a particular gene, and my objective is to amplify that gene, extract it, and then later on use it for my own experimental purposes. So, for doing that, the technique that one uses is polymerase chain reaction or PCR reaction, and that is what we are going to demonstrate today. So, in order to set up a PCR reaction, a polymerase chain reaction, there are a few components that you need to have at hand. So, you will require nuclease free water, you will require your template DNA. For today's experiment, as I had mentioned, I will be using cDNA isolated from the spleen of mouse. So, that will act as my template DNA. I will also use two primers, a forward primer and a reverse primer, with which I will specifically amplify a single particular gene of interest from my template DNA. And most importantly, I will need a PCR master mix. So, basically the PCR master mix will contain a DNA polymerase. For today's experiment, the master mix that I am using contains Taq polymerase. Taq polymerase is a polymerase that has been isolated from a thermophile bacteria, Thermus aquaticus. So, the special property of this particular polymerase is that it is stable even at high temperatures, because as you will be knowing from your theory classes, during the polymerase chain reaction, the enzyme periodically has to cycle through very high temperatures greater than 90 or 95 degrees centigrade. So, normal polymerases might lose their activity at such high temperature, but Taq polymerase remains functional during the PCR reaction. Apart from Taq polymerase, the PCR master mix also contains 4 types of DNTPs. It contains magnesium chloride, and all this is suspended in a suitable buffer, usually tris buffer. So, to summarize, in order to set up a PCR reaction, you need to have a template DNA, you need to have your forward primer and reverse primer, you need to have your PCR super mix, and to make up the volume to the right amount, you need to add nuclease free water to your reaction mixture. Be careful while you are using water, do not just use any source of water. Make sure that your water does not contain DNAs or RNAs. So, normally what we use is type 1 deionized water, which has been specially treated so that the DNAs have been already removed. So, the first step when we are setting up a PCR reaction, at the very beginning what you must do is wear a fresh clean pair of gloves, because PCR is a very sensitive reaction and you do not want to contaminate your sample with nucleases. Do not use your bare hands while setting up this reaction, because even the sweat in our hands, they contain nucleases, they could damage and negatively impact your experiment. And so, to summarize, do not use used gloves. Suppose in the lab you used a pair of gloves to clean your bench top, and then wearing those you came to set up a PCR reaction. Try to avoid that. Gloves are not expensive. Each time you set up a new reaction, wear a fresh pair of gloves. After you have done the PCR, you can use these gloves for some less sensitive purposes, but to emphasize, for PCR, new gloves always. Once you have prepared yourself in this manner, the next thing that you need to do is you need to thaw all the reaction components in ice. So, keep in mind that your PCR master mix, your template DNA, all your primers, they are all stored in -20 degree centigrade, and Most importantly, the PCR master mix that you have purchased or prepared in your lab, the main vial or the main stock is stored in -20, and a good practice is to make small aliquots of say 150 microliters in different tubes. That way you do not freeze thaw your entire amount of master mix at one go. Each time you set up a reaction, you take one tube and you put it in ice. You do not roam around with it in your hand, you put it in ice, and by keeping it in ice you thaw it gently and then you set up the reaction. So, one thing that I recommend before setting up your PCR reaction, what you should always do is write down the components that you will be mixing together in your laboratory notebook, and each time you add a component just put a tick, because keep in mind that in the molecular biology lab we mostly deal with colorless liquids, and today I am going to be demonstrating the setting up of a single PCR reaction, but while working in the lab you will see that many times you need to set up simultaneously 5 or 6 different PCR reactions. At that time it is very difficult to lose track of how much amount of which component has been added in which tube. So, for such cases just write down what you are adding, and each time you add something put a tick. This will help you while working in the lab. So, while mixing the different components of the PCR reaction, keep in mind that the order in which you add the components is also quite important. So, usually today I will be setting up a 50 microliter PCR reaction, and the first component that I am going to add is 20 microliters of nucleus free water. I have written down the recipe and you will be shown the recipe in a subsequent slide. So, I am taking the nucleus free water from the ice bucket, and now I am going to pipe it out 20 microliters and add it to my PCR tube. So, PCR reactions are small volume reactions. We only usually set them up in either 25 microliter volume or 50 microliter volume or 100 microliter volumes. So, you will notice that PCR tubes are much smaller in size than our regular centrifuge tubes, and while pipetting into PCR tubes, keep in mind that it is very easy to form bubbles and it is not good practice to form bubbles. So, pipette things very carefully, take your time, be slow, but make sure that you have as minimum number of bubbles as possible. With practice you can end up mixing everything without forming any bubbles. So, first we added 20 microliters of our nucleus free water. Now, I am going to add my template DNA. So, I am going to take 3 microliters of template DNA. In this case, for today's experiment, it is cDNA isolated from the spleen of a mouse. So, I am going to measure 3 microliters of template DNA and add it to my PCR tube. So, after adding the nucleus free water, next what I am going to add is the template DNA. For today's experiment, I am using cDNA isolated from the spleen of mouse as the template DNA, and I am going to add 3 microliters of that to my PCR tube. Keep them back in ice, take it up. Yes. Be careful so that you mix them properly, but at the same time you do not introduce bubbles into your PCR tube. Okay. I have added the template DNA, so I am just going to put a tick here, and after this I am going to add my primers. I will begin by adding 1 microliter of my forward primer, and once I have added that, I am going to add 1 microliter of my reverse primer. So, first I am going to add my forward primer, just you do not need much of it, 1 microliter is sufficient if you have a 20 micromolar working solution of your primers. So, yes, I have added the forward primer. I will just put a tick, because see, while working in the lab you will notice that I am adding very small volumes. So, even by seeing the tube it will not be possible to understand whether I have added 1 microliter or not. If I was adding say 500 microliters to a particular tube, I could look at it and say okay, I have or have not added. But since we are working with very small volumes of colorless liquids, add them and put a tick in your laboratory notebook. That will help you avert many mistakes. So, I have added the reverse primer as well. Just discard the tube, release my pipette, and yes, once it is released, just put a tick. So, till now I have added nucleus free water, the template DNA, my forward primer and reverse primer. The last component that I am going to add to my PCR tube is my 2x PCR master mix. Since I set up a 50 microliter reaction, I want to set up a 50 microliter reaction, I am going to add 25 microliters of my 2x master mix. So, just set up my pipette and now we are going to take the PCR master mix. When we had taken it out from the -20 refrigerator, it was thawed on ice. I am going to put it back on ice again, and now I am going to add this to my PCR tube. Okay. Now that I have added it, before proceeding further, I need to make sure that all the components have been properly mixed together. So, I am just going to pipette the whole mixture up and down a couple of times. Be very gentle, it is very easy to introduce bubbles at this stage, and it is not good lab practice to have bubbles in your PCR tube. Careful. So, I have thoroughly mixed together all the different components of the PCR reaction in the tube. You can pipette it. Sometimes some people prefer centrifuging the tube once just to mix together everything thoroughly. You can use whatever technique is comfortable with you. And once that you have added everything, just put your PCR tube in ice until and unless you are ready to set it up in the thermocycler machine. So, we have mixed together the different components for setting up the polymerase chain reaction, and we have kept the PCR tube in ice. So, now I have switched on this machine. So, this machine is a thermal cycler, and now I am going to set up a particular reaction condition under which the PCR is going to take place. So, as you can see here, there are 3 stages in this particular PCR reaction that I have chosen today. In stage 1, for 3 minutes, the temperature will be made at 95 degrees at the very beginning of the reaction. After that, the reaction will move on to stage 2. So, stage 2 is where the actual amplification of our gene of interest will occur. So, what will happen is, initially for 45 seconds the temperature will be kept at 95 degrees, after which the temperature is going to be brought down to 56 degrees centigrade for another and be maintained there for another 45 seconds, and then finally the temperature will be raised to 72 degrees centigrade and maintained for 1 minute. This entire pattern or triplet of temperature cyclings constitutes one entire PCR cycle. This stage 2 will be repeated 35 times. So, I have 35 such cycles. So, to summarize, initially for 45 seconds we maintain the tube at 95 degrees, then we lower the tube to 56 degrees and maintain it for another 45 seconds. Then the temperature is slightly raised to 72 degrees and maintained for 1 minute. After that, again the temperature is raised for 95 to 95 degrees centigrade for maintained for 45 seconds, brought down to 56 degrees centigrade, maintained for 45 seconds, raised to 72 degrees centigrade, maintained for 1 minute, and again the third cycle begins as I have described. So, 35 such cycles is what I have chosen for today's PCR reaction. So, at 95 degrees centigrade what happens is basically your double stranded template DNA becomes single stranded. The double strand opens up. At 56 degrees what will happen is that the primers will anneal to their complementary sites, and at 72 degrees what is expected to happen is that primer elongation will take place, which is why we need 3 different temperatures, and this is why this machine is commonly called as a thermal cycler. It can quickly change temperatures in very short spans of time from 95 to 56 to 72, and it keeps on doing it repeatedly. After 35 such cycles have occurred, we will move on to stage 3 of our PCR reaction. It is the final stage where we are going to carry out elongation at 72 degrees centigrade for one last time for a prolonged time period. I have set up for 7 minutes. After that, the machine will bring down the temperature to 4 degrees centigrade, and it's written that it will be maintained for infinite. So, basically what means is that after the reaction is over, the tube will be maintained at 4 degrees until and unless you take out your sample and switch off the machine. So, we have a 50 microliter PCR reaction, and this is the template that I am going to be using. So, now I am just going to choose the block. So, we have 3 different blocks, which means we can run 3 independent PCR reactions simultaneously. Today I am going to run only one. So, it is in block 2. I will just raise it. I will take my PCR tube from ice and put it here. So, it is very simple, just put your tube back here and carefully bring down the lid. You need to be careful. So, I have set up the reaction, and now I am going to take my PCR tube in this block. I will put my PCR tube here, and you need to be very careful. The inner surface of the lid of this block is actually very hot. Do not touch it, you might end up burning your fingers. Then you bring down the lid. So, basically in this thermal cycler we can set up 3 different PCR reactions simultaneously. Today we are going to do only one. Now that I have put my PCR tube in the thermal cycler block 2, what I am going to do is I will start the PCR reaction. So, it is beginning. Excuse me. So, as you can see now, it will take around 2 hours for the reaction to complete, and the temperature is rising. I will just show you what is happening. So, at this state right now the temperature is being raised to 95 degrees, and subsequently we will see that for the next 3 minutes it will be maintained at 95 to cover stage 1. Stage 2 is the stage at which actual amplification of your gene of interest will occur. So, for 35 times it will keep on varying the temperature from 95 to 56 to 72 repeatedly. So, these temperatures have been decided upon depending upon the experiment that one wants to do, and the machine is showing that after close to 2 hours the reaction will be done, and they will store the PCR tube at 4 degrees until and unless I come and retrieve the tube and switch off the machine. So, our PCR reaction is over. What I am going to do now is remove my sample from the machine. Take out the tube, this is back, So keep it here. So now what I need to do is I need to check if at all my gene of interest has been amplified or not, and the way we do that is I am going to take 5 microliters of this sample and I am going to run it on an agarose gel, and if I see any bands corresponding to the length of the gene or PCR product that I am expecting, it means that the PCR reaction has occurred successfully. After PCR amplification of the gene of interest, we will next move on to the double digestion step. However, prior to that, what we need to do is we need to remove the excess or unused primers that were used during the PCR, or we need to remove the different enzymes that are present, and the way we do that is we use commercially available kits known as PCR cleanup kits, which help us to isolate or extract only the DNA and get rid of the small primer fragments as well as enzymes and other impurities. After that, what I am going to do is, now I am going to set up a double digestion reaction. So in order to do that, I am going to take 40 microliters of my PCR product that I have. I'll just set the pipette at 40. So basically I am setting up a 50 microliter double digestion reaction. Depending on the concentration of DNA that I have. So I am going to need for today's reaction 40 microliters of DNA. If you remember, I had set up a 50 microliter PCR reaction. I had used 5 microliters for analyzing it on an agarose gel, and I still have sufficient amount for digesting. So I have now taken 40 microliters of the DNA. Next I am going to add water. I will add 3 microliters of nucleus free water. Just set up the pipette. Added the water, and I already added the template, the amplified DNA, and I have added water. Next I am going to add 3 microliters of the buffer in which my restriction enzymes are most active. Be careful to note that different restriction enzymes are efficient or active in different buffers. So always check the product information of your restriction enzyme and find out which buffer is ideal for showing 100% activity of the enzyme. So I am going to take that buffer and I am going to use 5 microliters of that buffer. 3.1 buffer, and I am going to add it into my digestion mixture. I have added DNA, I have added water, I have added buffer. So now I am going to add my 2 different restriction enzymes, 1 microliter of each, and yes, so restriction enzyme 1, I take 1 microliter of that. So when you are pipetting small volumes, always check that you actually have collected some liquid in your pipette tip. Often it might be that, especially when your tube is almost empty, it might be that you think you have taken out something but you don't, and then if you miss adding that, your reaction will not work. So when you are pipetting such small volumes, 1 microliter or 2 microliter, the best way is to look closely and see that you have some liquid in your tip and then proceed. So I have added my second restriction endonuclease. Yes. So just release my pipette before proceeding. Always release pipettes, do not store them under stressed conditions, because then you will damage your instrument. So I have added both my restriction enzymes, and thus I have set up my 50 microliter double digestion reaction. So what I am going to do is, now I am going to incubate this at 37 degrees centigrade for 1 hour. So how long you need to carry out digestion also depends on the type of enzyme that you are using. Some enzymes have been modified or enhanced so that they can work in a quick manner, and some enzymes need a bit more time. Depending on the enzymes that you are using, check the information. For today's experiment, I am going to set up a digestion reaction which is going to continue for 1 hour, and it's going to take place at 37 degrees. So I am going to basically keep the digestion reaction in a water bath. So I have now set up the digestion reaction for the insert. Keep in mind that you also need to digest your vector into which you want to clone your gene of interest. So I have also done that. I am not showing that to you, but similar to this, I have also set up a double digestion reaction for digesting the vector into which I want to clone my gene of interest. And I am sure you know from your theory classes that for cloning to occur, you need to make sure that both your gene of interest as well as your vector have been digested with the same pair of restriction enzymes. Now we are just going to set up those tubes in the 37 degree water bath. So now we are going to digest both our vector as well as insert. We have already added the restriction enzymes and set up the digestion reaction. Our enzyme works best at 37 degrees when kept for 1 hour. So we are going to use a 37 degree water bath. As you can see, the temperature of the water inside this water bath is maintained at 37 degrees centigrade. And now what I am going to do is, basically I will take my 2 vials containing my vector and insert set up for digestion and keep it here in 37 degrees centigrade for 1 hour. So the digestion will take place for 1 hour. So after digesting both the vector as well as our PCR product with the same set of restriction digestion enzymes, what we did was we ran them on an agarose gel, and after digestion, see, no process is 100% efficient. So there is always a possibility that some amount of your vector is undigested, while usually a larger portion of your vector is digested by both enzymes. So the vector that I am using today, if it is undigested, then it has a size of approximately 7 to 8 kilobases, but if it is digested by this particular set of enzymes, then the portion which gets removed has a size of around 2 kilobase and the remaining vector is 5 kilobases. So what we did was, after digestion we ran the vector on a gel, and after analyzing the gel, the band which corresponded to the 5 kilobase region, which is our digested vector, we just cut out that portion of the agarose gel and melted the gel and extracted the digested vector DNA from there. So after setting up digestion in a tube, you do not use the entire tube contents for ligation. You first purify and extract the digested portion of DNA by running it through an agarose gel and cutting out the region which corresponds to your digested product. So, similarly, not only the vector, but also the PCR product of interest has been digested and then purified by running it through a gel and cutting out only the band of interest. So, I have already done that, and now what I am going to do is I am going to set up a ligation reaction. So, for that I am going to first take, so the recipe, we will provide you with the recipe elsewhere. I have just written it down while I am doing the reaction, so that I can keep track of what I have added. So, first I am going to take 11.5 microliters of nuclease free water. Yes. So, I will take 11.5 microliters of nuclease free water. Careful while pipetting. Next, I am going to take, I will just release the pipette. Next, I am going to add my buffer. So, keep in mind ligation, the ligase that I am going to use today, T4 DNA ligase, it comes with a special buffer which contains ATP. So, ligation is an ATP dependent process. Therefore, the buffer that you are going to use contains ATP, and you should always store it in ice, because if the buffer goes bad, the ligation reaction will not take place. So, I am going to just take 2 microliters of that buffer now. Let me just set up the pipette. Yes. So, 10x buffer, I will just take it out of ice, pipette out 2 microliters and put it back on ice and add the buffer here. So, I have already added the water and I have added the buffer. Now, I am going to add 2 microliters of my double digested vector. So, I will take 2 microliters of my double digested vector and add it to my reaction tube. Also, as I keep adding, I just ensure that things are mixed properly by pipetting it a couple of times. I have added, excuse me, I have added my vector. Now, I am going to add 4 microliters of my PCR product, which has also been digested by the same set of restriction enzymes. Now, keep in mind that volumes here do not have much meaning. Basically, the way these reactions are set up is that you, or I personally prefer that you should have a 1:3 molar ratio, where you have 1 part of digested vector and you have 3 parts of double digested insert. So, you have excess double digested insert compared to your vector. You can also set up a 1:5 or even sometimes 1:1 ratio of vector and insert in your ligation reaction. So, till now I have added the nuclease free water, I have added the 10x ligation buffer, I have added my double digested PCR product and my double digested empty vector. So, now I am going to add the last component basically that is left for the ligation reaction to be initiated. I am going to add 0.5 microliters of ligase, T4 DNA ligase. I am using T4 DNA ligase for today's experiment. So, I will just take a very small amount, 0.5 microliters, add it to my ligation mixture. So, we can pipette it up down a couple of times. I will just increase the volume and I will mix it thoroughly. And so, the ligation reaction is going to take place at room temperature for half an hour. So, we will just leave the ligation mixture in a tube on our lab bench at room temperature for half an hour, after which we are going to proceed with our experiment.